Today it's my pleasure to introduce you to Rhiju. He actually is local to Stanford, and he actually did his PhD work here, I guess in particle physics and cosmology, and then he did a postdoc at Washington University where he participated a little bit on Foldit, crowdsourced protein folding game, and then recently, I guess in the last 6 years, came to Stanford to work on a game where he is able to basically use the feedback from thousands upon hundreds of thousands of players to figure out the best way RNA molecules can fold and design new structures. And these molecules are relatively important to all of your biology, as you'll learn soon enough. So my pleasure, Rhiju.
So first of all, thanks to the organizers of the game series for inviting me to speak. And yeah, today, I hope you won't mind, I'm going to tell you about this slightly bananas project. It started out as a side project that I kept hidden from my faculty colleagues because I thought it was sufficiently weird to invite tens of thousands of gamers to use the biomedical research tools in our lab. But nowadays, I'm tremendously excited to tell you about how these folks have taught me a huge number of new things about our research in RNA medicine and how I think they're going to have a potentially large impact on the science of the future. So RNAs are these, let me get a laser pointer. RNAs are chain-like molecules that play critical roles in every major process of biology and in many diseases. Brain diseases, like spinal muscular atrophy and many forms of Parkinson's, can be traced to defects in how RNAs fold up in neurons. And the H1N1 flu and the HIV virus are RNA viruses. They harbor critical RNA elements that are important for replication and for infection. Okay, so my lab and many of their labs around the world are trying to defeat these diseases by targeting the RNAs involved and by refolding them. And some of us want to fight fire with fire. We want to, what if you could disrupt or defeat one of these RNA diseases by making devices that themselves hold RNAs at their heart? So there are many challenges in RNA medicine, but in the middle of all of these efforts, at the core of all these efforts, is a basic biophysical puzzle that remains unsolved. So I'm showing you here a structure of a single RNA strand, and you can imagine that this is the shape that I want the RNA to fold back into, to be at the heart of a device that might sense a viral infection in the cell and then initiate an antiviral response. Now, the problem is this. The RNA design problem is to fill in these blanks with A, C, G, and U, the 4 letters of the RNA alphabet, so that A's pair with U's and C's pair with G's. And the trick here, the thing that makes it a hard puzzle is if I put an A here against a U, I want it to pair just with that U and not with a U elsewhere in the structure. In other words, I have to prevent a misfolding of the sequence when I make it in the test tube. And that large set of constraints of avoiding misfolding is what makes this problem analogous to, say, a Sudoku or a crossword puzzle, and makes it challenging not just for a human, but also for a computer. The thing that makes it really difficult is that these RNAs don't fold up properly when you make them in a test tube, much less in a cell. And I'd like to illustrate that with a little quiz here. I'm showing you here 2 sequences that have been designed to fold up into that same structure. And I'm going to ask you to guess which of these sequences folds up correctly and which one doesn't. We now know that one of them folds up correctly, one of them doesn't. Now, before you vote, I'm going to tell you a couple of things about the sequences. So first of all, if you put either of these sequences into the best available computational structure prediction programs for RNA, they both pass with flying colors. They're predicted to be extremely stable at room temperature with very low probability of misfolding to alternative folds. So that's no help. But they are different, okay? So if you look here at the left-hand one, it has more of these yellow or A residues in the unpaired regions, whereas the one on the right is a little bit more diverse. So now I'm going to ask you to just guess which of these folds up correctly. And just humor me and vote for one. Who thinks this left-hand one folds up correctly? Okay, I got 1, 2, 3, 4, 5 people. Okay, who thinks the right-hand one folds up correctly? Okay, so the majority of people go for this one. So I should tell you, I didn't know the answer to this puzzle, and no computer did either. We had to turn to nature to give us the answer, and it's the left-hand one that's correct, okay? And usually when I use the word diverse for the right-hand one, it's what triggers people to go over to the incorrect solution. But let me tell you how we know this. We carry out a method called high-throughput chemical mapping, which can label which of these letters of the RNA in a test tube are exposed to solution and therefore unpaired in the fold. And we've actually colored these RNAs by those data. Yellow means chemically reactive or unpaired, and blue means unreactive or likely paired. And this match of unpaired and paired regions is about as good as it gets for the one on the left. But for the right-hand one, well, these long appendages coming off the fold, they look okay. They have these yellow fingertips and yellow knuckles. But the middle ring of the structure has a chemical reactivity pattern that's more consistent with a slightly alternative fold. It's a small error, but even a tiny error like this could totally throw off an RNA therapeutic. Now, there's an extra twist to this quiz, which is the source of the designs. Okay, the one on the right, the incorrect one, came from the most sophisticated computational RNA design algorithm that we have. It's called NUPAC. It's been developed over more than a decade at Caltech. The one on the left, the correct one, was named Taipan, and it was created by a video game player named Penguian, who had no prior expertise in RNA folding or design. So this is an example of an anecdotal example of a human beating a computer or an expert. And there's a precedent for this kind of result in more than a decade of scientific discovery projects. Okay? These projects really began in the astronomy community with NASA clickworkers and the many Zooniverse projects. These games have successfully crowdsourced the classification of telescope images to thousands and now millions of volunteers on the internet. In 2008, the first of these video games for biochemistry came out. It was called Fold It, and it was developed at the University of Washington. And you may have seen press a few years ago that a small group of Fold It players were able to unlock the structure of a retroviral protease in a data set that had remained unlocked for the previous decade in the hands of expert structural biologists. So what I want to tell you about today is a new project in this lineage called EteRNA. EteRNA is unique in its inclusion of actual experimental bioscience as part of the gameplay. So unlike Fold It, which challenges its players purely to solve puzzles in silico, Fold It challenges its players to look at prior experimental data and then to design new experiments. The tagline of EteRNA is played by humans, scored by nature. Let me show you how it works. Every week we've had a lab challenge shape, and players have access to a mildly addictive game-like interface to send us sequences that they think will fold up into these shapes. And then they vote on which ones they like best after publishing their solutions to each other. And then we synthesize these solutions in my lab at Stanford, return back the experimental data as this yellow and blue coloring back to the players. And if they haven't solved it, they have another crack at it next week. And if they have solved it, they go on to a more complex shape in the next round. So I want to emphasize something here. EteRNA is not just a video game, although that's a very important part of it. My lab at Stanford, actually, I burned through my whole startup synthesizing these random sequences from God knows who on the internet in an effort to give back experimental feedback to the players. And you can see here what we're trying to do is we're going from hypotheses to results, experimental results, to analysis of those results to the next round of hypotheses. EteRNA was exciting to us because we were trying to crowdsource the entire scientific method on the internet scale. So because this is a game seminar, I'm going to give you a little tour of what the puzzle looks like, or what the game looks like. And then I'll show you the experimental results. So we developed this game back in 2010, 2011. And it has many of the core components you might hope for in a game. There's at least 3 things that I want to point out that I learned about from my computer scientist colleagues who wrote the game. Okay, first of all, because it's putting people into a rather different molecular world, there are many elements that are pedagogical. So here's a tutorial that tells you about how RNA folds. It tells you about the elements of the game, like these stacks. It tells you about the different kinds of base pairs. And then it gives you a mission. Okay, so it's pedagogical. Secondly, as you start playing, you're designing a sequence. Because it's a scientific game, it should have a reasonably realistic scientific model. This is using the so-called Vienna fold model, which has been developed over the previous about 15 to 20 years in the RNA field. It's not a perfect model of reality, otherwise we wouldn't need the game, but it's the best one we have. And players get immediate feedback on whether their sequences are folding, at least with this imperfect computational model. So in addition to being scientific, the third thing that the game has is it has a kind of many layers of feedback to the player, including positive feedback. So One of the more subtle things that I only discovered later in the game was that if you move your mouse around these bubbles, you'll see that they'll move. This is something you don't register consciously, but it subconsciously makes you feel good about being in the environment. And then the game, of course, has elements of juiciness, excessive positive feedback when you do something useful. So actually, if I solve the puzzle, you'll see here that we'll get a huge number of bubbles and lights and sound effects, which are not coming out right now, and then you're done. OK, so after you go through these sets of puzzles, if you earn 10,000 points, you earn the privilege of playing the lab puzzles. And I'm not going to show you how to solve one of the lab puzzles, but I will show you what happens as you start solving it. You then get to see everyone else's solutions. And this is an important part. If there's already been a round of the puzzles, you can see previous player solutions. They're ordered by their score here. And those yellow and blue colorings are the ones that I showed you in the slides. Those are actual experimental data. You can then take a quick look at prior player solutions along with their commentary on what they did to prepare those sequences. And then you can actually see those colorings in the same kind of in-game representation that you use to then design the sequences. So let me just, if my internet's working, you'll be able to see it. Yeah, there you go. And so here's the player, here's the puzzle. In addition to having what the target solution was, you can actually get a mode that tries to resimulate the structure of the RNA given the experimental data. In this case, it's actually a close match, so there's no change in the fold. OK. So now let me show you what EteRNA players did in the first year of the project. So to start with, for each of the RNA shapes here, as a baseline, we synthesize designs that are made by these prior computational algorithms. And I'm not showing you the full data sets here, but just a summary score going from 0 to 100, where 100 is best. And these gray symbols are these computational designs. And about half of them are kind of in this B plus A minus range. About half of them are in this failing range. None of them are close to 100, which is the best score you could possibly achieve. How well did the players do? Well, they did about the same as this fairly poor computational algorithm. And you might expect this. This is actually the very first round of EteRNA, and the players hadn't yet gotten experimental feedback. But you hope that players can learn from these data. You hope that they'll evolve to get rules for new puzzles. And here are the results over the next 6 to 8 months of the game. I'm going to point to two trends in the data. First, the gray points are these solutions from computational algorithms. And you can see they're getting almost monotonically worse as the shapes are getting more and more complex. In contrast, the human players, their performance is increasing also almost monotonically, to the point that by these last two puzzles, the first time the players saw the puzzles, which are these blue symbols, their median score was better than the best score seen by prior computational algorithms. And about a quarter of their designs were indistinguishable from perfect, given our experimental assay. So how is this happening? So at this point in the project, I had these long arguments with Adrian Troy, my gaming collaborator. So he has a background in computer science, and he really wanted to record mouse clicks of the players and, classic Silicon Valley style, carry out deep learning and create a new amazing algorithm. And I was saying, no, that's too complicated. At some point, the debate stopped because we just started looking at the chat and the forums associated with the project. And people were just telling us what they were doing. Here's an example. This is from a player named Eli Fisker. Let me read you a strategy that he shared with the other players. And then I'll explain to you what it looks like on the diagram. All GC pairs in the multi-loop junctions have to turn in the same direction. Red nucleotide to the right, green nucleotide to the left. Exception, the GC pair connecting the multi-loop and the neck are allowed to turn in both directions without being penalized. Okay, so the first time I started reading these ideas, I had no idea what Eli and others were talking about, because there's no concept of turning in the same direction in the expert literature, or what a neck is. But finally, the players published a glossary of their terms. Okay? It's a jargon that had arisen in the game. And then I could understand it. This actually is pretty reasonable to say that certain loops would be stabilized with a certain so-called 5' 3' arrangement of CG pairs. And it's reasonable to allow there to be some flexibility in trying to design the most longest range stem, which is what they call the neck. It's reasonable, but I wasn't really sure if these are the strategies that the players are using to actually get good solutions. I had to take it seriously because Eli Fisker was the best player in the game. So he's actually the one who's producing these near-perfect solutions in every round. But at the same time, I worried that perhaps his own human intuition was confounding his success or his interpretation of what was leading to success in his designs. And so we then decided to create a new game within the game. It's called a strategy market. We rewarded players not just for submitting designs that would work, we rewarded them for finding patterns in the designs that were successful. And we rewarded them. We coded up their ideas, which were submitted to us in plain English, and we returned points based on how well those ideas explained prior experimental data. When we had a set of about 40 of these proposed rules, we then carried out a very simple machine learning to create an optimal combination of these scores that best explained prior data. And then we wrote a very simple trial and error algorithm that optimized this player-derived score. Okay? And we call this EteRNA. Now, so the point of EteRNA was to do two things. First of all, it was a way to test the player's rules and intuition in a way that didn't involve the players themselves confounding their design process. And secondly, if EteRNA succeeded, this would be a chance to distill the collective intuition of a very large community of folks into an algorithm that other people could use in the RNA engineering community. So we then tested EteRNA on 9 other puzzles that were completely different from anything that the players had seen before. Okay, I'm going to show you results on one of these right now. This is the shape, again, that had previously been untested in EteRNA. I'm showing you here the results from 2 prior computational algorithms, and the players also got a crack at it, and as usual, they're beating the prior computational methods. How well did EteRNA do? You can see it bridged the gap between the prior methods and the players themselves. And indeed, on all 9 of these independent puzzles, well, in the majority of cases, EteRNA bridged the gap between the prior methods and the players themselves. In all cases, the best score from EteRNA was better than the best scores achieved by prior computational algorithms. So this was super exciting to us because this attempt to distill the ideas of a lot of smart folks responding to experimental feedback, it's really the best algorithm we have in designing RNA structure, and it's now going into routine use in my lab and other labs in the community. So at this point, I thought, okay, look, I've run out of startup funds. It's time to sunset the project. And I got some help from folks here at MediaX, actually, to keep the project going. I thought, okay, maybe we should just sunset the project now. It's mission accomplished. But around that time, the players started sending us some really amazing ideas. Okay? So as an example, El Rapi was talking to me in chat. And he said, I like the idea of shapes and constraints that help us discover where the model does not work. And so what he had been doing, I realized, is for all the puzzles, he'd been submitting sequences that only used 2 of the 4 letters of RNA, these yellow and blue A and U letters, and with the explicit prediction that even though these fold up perfectly in a computer, they will fold up imperfectly when we synthesize them. Okay? So this is a way to incisively fix the models, the computational models, for one sector of the RNA alphabet. Then there were players telling us, oh, your shapes are too easy now. You should be giving us challenges that look like these snowflakes or pinwheels. And it turns out these shapes, although they're quite small and should be really easy to design, they cannot be designed with any prior computational algorithms, even in silico, okay? Much less folding up in a test tube. But humans can solve them, at least within the EteRNA game. And there were players who were submitting us things that looked like very complex shapes, even things that looked like art. This came out a few months after Steve Jobs passed away. And I would love to know if these RNAs misfold or fold in the way that the players designed them. But I couldn't test them, okay? We don't have the scores for these designs. And the reason why is a bottleneck that had been there since the beginning of the game, which I haven't told you about. You see, at the time we wrote up our first paper, which was published last year, and we're thinking about finishing up the game, we had 80,000 registered players. About 6,000 had played enough to earn the privilege of submitting us designs that we have synthesized. They had entered the lab. And we were getting about 1,000 designs per round. But my lab could only afford to make about 32 syntheses per round. So we're potentially missing out on hundreds of really interesting ideas during every experimental cycle of EteRNA. And so this has been a problem from the beginning of the project. We had many top players complaining to us that, okay, the voting in EteRNA is a popularity contest. We should change the voting system so that we'll elect a senate of players to go through all the designs and pick the ones that we'll have in every synthesis. There were some players saying we should have it just as a pure lottery system. There were players who were suggesting that we have a major league of really elite players and a minor league of not so good players. And I was about to, being in the scientific community, I was about to instill peer review, where every player had to look at 5 other designs before they submitted their design for synthesis and rank the other ones. And so this seemed like a impenetrable problem. It's a basic problem of political science where you have lots of people and very few resources and have to share them. But being in the Bay Area, I'm influenced by the techno-utopianism that is rampant in this area. And I thought, well, okay, maybe there's a technological solution for all this. And indeed there is. Okay? So some of you may know about the revolution that has been sweeping through the life sciences, where there's been a Moore's law, or actually a super Moore's law, in the availability of nucleic acid synthesis, like DNA and RNA synthesis, and DNA and RNA sequencing. Okay? So to the point where I think if you go into the clinic here over the next couple of years with diagnosed with a chronic disease, you and your disease, their genomes will be sequenced. Okay? So this has been happening over the last few years, and we realized that we could take technologies to print glass slides with arbitrary DNA sequences to print thousands of sequences that EteRNA players were typing into our game. And we also realized we could develop a version of our chemical mapping experimental technology to then probe how these RNAs fold in a massively parallel manner. I'm not going to go into all the details here, but the upshot was that by the end of 2013, every sequence that was being typed by a top player into the game was getting synthesized. Okay? And so we started returning these data back to the players in the game at a huge rate. They were solving these lab puzzles at a fantastic clip. We started to realize that maybe there's a better way to harness this really radically increased experimental throughput. And so what we've been playing with for the last year and a half is a new kind of gameplay in EteRNA. So now, all of the puzzles in EteRNA, they don't come from me or people in my lab. We incentivize players to come up with complex hypotheses about RNA folding and design, to team up with each other and propose the test of these hypotheses. And we reward them for coming up with experiments that might test them, or puzzles that will elicit designs from other players that will test those hypotheses. And as if they're voted in, if their projects become the lab puzzle of the round, then we carry out their experiments in this massively parallel new pipeline. And then there's an additional thing. I've already told you that players are coming up with the hypotheses and devising experiments to test them. The last part of this gameplay is we ask the players to create tiny papers, micro publications, explaining what they did. And if they complete these, then they get extra points to get into the next round. Now, those of you who are, say, graduate students here at Stanford and are here in the audience will know it's usually pretty tedious to go and write up your paper. But this is not so hard because as the entry fee for playing EteRNA, the players have already given a title to their projects. Their introduction is their hypotheses. The methods, well, we are doing the experiments for them and providing, and the results are also provided within the game. So all the players really have to do is come up with a conclusion for what they did. And that's it. That's their micro-publication, and they get the points for the next round. And so since we initiated this new kind of gameplay, we call it the Cloud Lab game, players have been going bananas. So there are some players who are working with, for example, biologists at UC Santa Cruz and at Stanford to go look at the folds of every possible transfer RNA in the cells in the human body with the hypothesis that many of them misfold unless they're modified by certain modifications. There are folks who are designing these frog leg type motifs which they want to put inside new kinds of molecular motors. There are also players who are going after really esoteric things like these. They want to create these zigzag and zagzag motifs. This is a motif that players discovered is very tiny but not solvable by current computer algorithms, but the players have found rules to try to make them. And the overall upshot is, as of about a month ago, we have synthesized over 40,000 sequences in 379 projects. And these are all devised by players themselves and not by the so-called expert scientists. So the last goal here, and we're now taking the first steps toward it, is to incentivize the players to collate their results into publications that they could submit for peer review. Okay? And so I just want to not totally oversell this. I mean, the goal here, none of these projects are going to cure cancer in and of themselves. But suppose that a Stanford researcher or an MIT researcher in the next year or two realizes that she needs to package silencing RNAs into a spiral shape in order to deliver that as an anti-tumor therapy. Right now, if she Googles RNA spiral, she won't find any papers in the literature. It's just too esoteric of a subject. Our hope is that in a couple of years, she'll Google that, get a paper that explains how you do it and how not to do it, backed up by real experimental data. Okay? And you can do a back-of-the-envelope calculation that Okay. There are only maybe a dozen labs who work on basic principles of RNA folding and design around the world now. Okay? But if we can get this publication pipeline going with EteRNA players, there's a chance that we can create experimentally validated knowledge at a rate that's comparable or maybe even exceeds that of all current labs combined. So before I take questions, I want to thank the other folks who are working on EteRNA. Adrian Troy and Ji-Hyung Lee, both at Carnegie Mellon University, with frequent visits to Stanford. And they're the ones who set up the game originally. What I forgot to put here is over the last year, as they've been finishing out in the project, their roles have been taken up by two extremely experienced software engineers, John Nickel and Fernando Portela, who we drew from the top 10 ranks of the game. So it turns out that several of the top players are software engineers. I want to thank the experimental team, which has been Anne Cladwang in my lab. And Johan Andreasen is working with us to create even a newer experimental platform where instead of synthesizing and probing thousands of RNAs per round, we're now able to do 100,000 RNAs per round. In fact, we're at a point where we actually have more synthesis slots than we have active players, and we're seeking to expand the game through a mobile expansion over the next year or two. I'd like to thank funding sources. And finally, the players themselves, they're the ones whose hypotheses that we're testing. And I'm happy to take questions.